Transport across the membrane is separated into three different parts or three processes. This lecture is about simple diffusion, membrane transport that doesn't require any kind of energy and also doesn't require any kind of facilitation like with the help of proteins. I have down here a representation of diffusion. Diffusion is defined as the movement of substances from where they're in greater concentration to where they're in lesser concentration. Or another way you could describe diffusion is movement down a concentration gradient from where concentration is very high to where it is lower. Here we have a drop of dye being dropped into a cup of water. At this point, the concentration of dye molecules is high. Diffusion is illustrated by the movement of these molecules of dye from where they're in greater concentration to where they're in lesser concentration. And notice that no matter how long we leave this sitting here, the molecules will remain spread out and in what we call an equilibrium, where there isn't an area of greater or lesser concentration of the molecules. Also, inside of this cup of water with dye, the dye molecules are still moving. But equilibrium means that every particle is moving kind of randomly, and so there will never be any region of higher concentration or lesser concentration. That defines diffusion, but let's apply it to transport across the membrane. Remember that we have a phospholipid bilayer separating the interior of the cell from the exterior of the cell, or the intracellular environment from the extracellular environment. Imagine that membrane, and on one side of the membrane there's a substance in a great concentration, higher concentration. As long as that membrane is permeable to the substance that's higher in higher concentration on one side of the membrane, then that substance or molecules of that substance will travel from where it's in greater concentration to where it's in lesser concentration. It'll keep traveling, and also note that once there are particles on the other side of the membrane, they can still very freely pass to the side where they began. And there reaches a point that we call equilibrium, where the concentration of the molecules is the same on both sides of the membrane. That is, if the substance can pass across the membrane. Now imagine we have two substances, one in greater concentration over here, one in greater concentration on the left, but these two aren't in equal concentration to each other. If the membrane is permeable to both substances will move across the membrane until each substance is in equilibrium. These are two different substances, and so their equilibrium doesn't necessarily depend on the other's equilibrium. Now the membrane is permeable to some substances. So here on the left I have a scale of permeability, and this is centimeters per second of diffusion. Now small nonpolar molecules like dissolved gases easily pass from one side of the membrane to the other. They're nonpolar, so they're not repelled by the nonpolar barrier, and they're very small, so they can easily slip through the membrane. There are some small polar molecules that have to be uncharged, but they are polar, that can also slip across the membrane, and that includes water. Glycerol is somewhat permeable, but once you get to something bigger than the molecule of glycerol, permeability decreases dramatically. So I know that occasionally glucose can cross the membrane unaided, but for the most part we consider glucose to be, or the membrane to be impermeable to glucose. And certainly sucrose and any other larger polar molecules, any of the macromolecules, with the exception of smaller lipids. Lipids like steroid hormones, testosterone, estrogen, glucocorticoids like cortisol, they can slip from one side to the other. Now anything that's charged, like ions or any charged large molecules, absolutely cannot cross the barrier unaided. We'll talk about how these things do cross the membrane in the next lecture, but that has to be aided with a protein. So all membranes have this specific fluidity, and that determines then what they're permeable to. Some things can pass easily, while others do not. If a membrane is more fluid, you know that it's more permeable. If it's less fluid, then it's less permeable. Our membranes maintain a concentration of phospholipids so that this permeability is always the case. These are easily passed through the membrane. These are also passed through the membrane. And then, of course, these with help. So, actually, let me go back a slide. Let's say that this is glycerol. This is the plasma membrane, and this is the interior of the cell. Glycerol molecules can easily slip through this membrane, through the phospholipid bilayer, and will move back and forth until the glycerol molecules are in equilibrium or equal concentration on each side of the membrane. All of these things diffuse from an area of greater concentration to an area of lesser concentration, and there's really nothing stopping them. Now let's think about the diffusion of water, which is the definition of osmosis. Here I have a starting point where I've got water and two regions of aqueous solution separated by a membrane. Now this membrane is only permeable to water. It's not permeable to something larger, like let's say this is an amino acid, which is larger and also charged. In this scenario, the concentration of water is greater on the left than it is on the right. More space is taken up by these amino acids than by the water molecules. Another thing we can say about these two solutions is that this one is hypotonic, less concentration of solute, or the things that are dissolved in here, and this one is hypertonic, a greater concentration of solute. Now, what's going to happen in this case is that water is going to move to dilute. So water is going to move from the hypotonic solution to the hypertonic solution, or water is going to move from where it is in greater concentration to where it's in lesser concentration. But no matter how you describe it, this is the result. The movement of water is such that we will have a greater concentration of water on this side of the membrane. And now the two solutions are in equilibrium, both in the concentration of water and also in solute. Now, our cells can easily allow water to move across membranes. And so we have to keep a careful concentration of solutes in order to make sure that we don't have net water movement in either direction. Here again we've got a lipid bilayer, greater concentration of solute, lesser concentration of solute. Again, our membrane is permeable to water. Water moves from low solute concentration, hypotonic, to high solute concentration, hypertonic. Water moves from where it's more concentrated to where it's less concentrated, or it moves to dilute. If the outside of the cell was hypotonic and the inside of the cell was hypertonic, you can imagine water rushing into the cell. That's a situation where in animal cells, a cell would lyse. If water diffused into the cell in order to dilute an incredibly hypertonic interior, the cell would lyse. In the case of plant cells, lysis isn't a problem because of the cell wall and because the vacuole stores up all the water. This is why we store red blood cells. We don't store red blood cells in pure water. We have to store red blood cells in some sort of salt solution with a concentration that is isotonic, or in the same concentration of solute as the cytoplasm of these red blood cells. If we stored it in pure water, then cells would burst as water diffused into the cell. In isotonic solution, there's a net flow. The net is equal here. There's a flow in and out of the cell, but there's no change in solute concentration within the interior of the cell. Now imagine if The hypertonic solution was outside of the cell, and the hypotonic solution was inside of the cell. If a cell was in hypertonic solution, water would move out because there's nothing stopping it. And in that case, you get a shriveled up cell, or plasmolysis. And the same thing does happen in plant cells. Water leaving the plant cells causes this plasmolysis of the cell. You could see an example of this if you were to place a stalk of celery, 2 stalks of celery, one in pure water and one in salt water. Within a very short period of time, you'll pull out that stalk of celery from the pure water and it'll still be crisp. If you pull out the stalk of celery from the salt water, you'll find it's rubbery and it doesn't snap in half. That's because all the water has left the cells, and the water has now moved out into the solution into which the celery is stored. Plasmolysis will happen to either plant or animal cells. Now, I've said earlier that there are substances that have to pass the membrane, like glucose, for example. All cells need glucose in order to create ATP. ATP is required to survive. It's our energy packet, our cell's energy packet. Things like glucose, amino acids, or anything bigger can pass from one side of the membrane to the other, but it requires help. That's called facilitated diffusion, and that's the subject of the next lecture.